Welcome to the Human AI podcast. I'm your host, ChatGPT is our guest. I'm going to be asking ChatGPT about the liver and the kidney today. If you haven't watched part one, watch it. It's going to make sense because we're not starting from the beginning. And yeah, without further ado, let's kick it off. So, ChatGPT, we left off at a kind of important point. I was about to ask you, but I got tired. About the organization of the kidney, specifically the outer layers. So can you talk to us about that a bit, please?
Absolutely. The kidney's outer layers are mainly the cortex and the capsule. The capsule is a tough, fibrous layer that protects the kidney. Underneath that, the cortex contains blood vessels and nephrons, which are the functional units that filter blood. Everything inside is well organized to
Wait. So the cortex. Okay. So we have the cortex as one outer layer of the kidney. What about the medulla? This is the medulla an inner area, and you mentioned the capsule too.
Exactly. The kidney's cortex is the outermost layer where blood filtration begins. The medulla is the inner region, containing structures like the renal pyramids, which help with urine formation and concentration. And the capsule is a tough, fibrous layer surrounding the kidney, offering protection.
So you said the pyramids.
Yes, the renal pyramids. They're cone-shaped tissues within the medulla. They contain the loops of Henle and collecting ducts, which are crucial for concentrating urine.
Interesting.
It is. The way these structures work together to filter blood and create urine is pretty fascinating.
Why do we call it medulla?
The term medulla comes from Latin, meaning marrow or innermost part. It's used in anatomy for inner regions, like the kidney's medulla, because it's surrounded by the outer cortex.
Do we have other medullas?
Yes. There's the adrenal medulla, which is the inner part of the adrenal gland, and even the medulla oblongata, part of the brainstem.
Interesting. And you mentioned the renal pyramids. What about the renal pelvis? What does that do?
It's a funnel-shaped structure in the kidney that collects urine from the calyces and channels it into the ureter, which then carries it to the bladder.
What do you mean?
The calyces are small cup-like cavities. They collect urine from the renal pyramids and pass it into the renal pelvis.
Is it big? Is the renal pelvis big?
Not particularly. It's large enough to gather urine from all the calyces, but still fits snugly within the kidney.
Is it like the head of the ureter?
Yeah, you could think of it that way. It's where the ureter starts.
Okay. And now talk to us a bit about the nephron and the sections.
Sure, the nephron is the kidney's functional unit. It starts with the glomerulus, a tiny blood-filtering cluster. Then there's the proximal tubule for reabsorption, the loop of Henle for concentrating urine, and the distal tubule, which fine-tunes the filtrate before it reaches the collecting duct.
Interesting.
It really is. Each part has a specific role, all contributing to the kidney's ability to filter blood and maintain balance.
Okay, and what about the distal tubule and the collecting duct? What's going on there?
In the distal tubule, final adjustments to the filtrate happen, like reabsorbing sodium and water. The collecting duct then fine-tunes the urine's concentration before it heads to the renal pelvis.
Interesting. So we have cortical nephrons and juxtamedullary nephrons?
Exactly. Cortical nephrons are mostly in the cortex, while juxtamedullary nephrons extend deeper into the medulla. The latter are crucial for producing concentrated urine.
Can you explain what juxtamedullary means? What's the juxta part?
Juxta means next to or near. So juxtamedullary nephrons are nephrons located near the boundary of the cortex and medulla.
And most of them are cortical.
Yes, most nephrons are cortical, about 85%. The juxtamedullary nephrons are fewer, but essential for concentrating urine.
What about the afferent arterioles? Where are they?
The afferent arteriole brings blood into the glomerulus, the nephron's filtering unit. It's like the entry point for blood to be filtered.
Got it. And how does the structure of the cortical and the juxtamedullary nephron differ?
The main difference is their location and loop of Henle length. Juxtamedullary nephrons have longer loops of Henle that extend deep into the medulla, allowing for more concentrated urine.
Interesting.
It is. The kidney's design is pretty amazing when you dive into the details.
Give me just 1 minute, guys.
Sure, take your time.
And sorry guys, let's get back to it. Tell me again about the structure of the cortical and the juxtamedullary nephrons and how it differs then. For instance, if you could mention the peritubular capillaries and the vasa recta.
No problem. Cortical nephrons have shorter loops of Henle, mostly in the cortex. They're surrounded by peritubular capillaries, which reabsorb nutrients. Juxtamedullary nephrons have longer loops that extend into the medulla, with vasa recta capillaries helping with urine concentration.
Interesting. Okay, and now, do we have sympathetic and parasympathetic nervous system stuff in the kidney?
Yes, definitely. The kidneys are influenced by both systems. The sympathetic nervous system can reduce blood flow and thus urine production, while the parasympathetic system has a more minor role in regulating kidney function.
Interesting. And so in the sympathetic nervous system, do we have postganglionic fibers? What do we have?
Exactly. Postganglionic sympathetic fibers innervate the kidneys, mainly controlling blood vessel constriction, which affects blood flow and filtration rate.
So why are they called postganglionic?
They're called postganglionic because they extend from the ganglia, where the preganglionic fibers synapse, to the target organ, in this case the kidneys.
Interesting. And also they have fibers from the celiac ganglion.
Yes, fibers from the celiac ganglion also contribute to regulating kidney function.
What's the celiac ganglion?
The celiac ganglion is a major nerve cluster in the abdomen. It supplies sympathetic fibers to various organs, including the kidneys.
Interesting.
It ties everything together, helping coordinate the body's response to different needs.
And where does the parasympathetic nervous system come from? Is it efferent supply from the vagus nerve?
Yes, the parasympathetic supply mainly comes from the vagus nerve, though its influence on the kidneys is less pronounced than the sympathetic system.
Okay, and we have different things that have to do with the nerve. We have filtration, reabsorption, secretion. If you could kind of explain to us the differences, maybe also mention transcellular and paracellular transport.
Sure. Filtration happens in the glomerulus, where blood plasma is filtered into the nephron. Reabsorption is when useful substances are taken back into the blood. Secretion is the transfer of additional waste from blood to the nephron. Transcellular transport involves movement through cells, while paracellular transport occurs between cells.
Interesting. And What about what's the tubular fluid?
Tubular fluid is the filtered plasma that flows through the nephron. It changes composition as substances are reabsorbed and secreted, eventually becoming urine.
Okay. So, when it comes to glomerular filtration, proteins and red blood cells are the main ones that don't go further, right?
Exactly. The glomerular filtration barrier is designed to prevent large molecules like proteins and cells, including red blood cells, from passing through. It's made up of 3 layers: the endothelium, the basement membrane, and the podocytes. These layers act together to ensure only smaller molecules like water, electrolytes, and waste products pass into the filtrate.
Okay, and let me ask you about tubular reabsorption. So, a lot of the water is filtered back, right?
That's right. The kidneys reabsorb about 99% of the filtered water. This process mainly happens in the proximal convoluted tubule. The rest is fine-tuned along the nephron, especially in the loop of Henle and collecting ducts, depending on your body's hydration needs.
Yes, what about sodium and chloride?
Sodium and chloride are also heavily reabsorbed. Most sodium reabsorption occurs in the proximal tubule, with fine-tuning in the distal tubule and collecting ducts. Chloride typically follows sodium to maintain electrical neutrality, so their reabsorption is closely linked.
And how does the sodium-potassium ATPase transporter play a role in all of this?
The sodium-potassium ATPase pump is crucial. It actively transports sodium out of the cells lining the nephron in exchange for potassium. This creates a concentration gradient that allows sodium to be reabsorbed from the filtrate back into the blood.
But I wonder, for potassium, we don't need potassium?
Oh, we definitely need potassium. It's essential for many bodily functions, like nerve signaling and muscle contractions. The sodium-potassium pump helps maintain the right balance by pumping potassium into cells, ensuring we have proper levels inside and outside our cells.
Yeah, but the fact that we have to get potassium in to get sodium out means that it's like we're bound to have a deficiency, unless I'm not understanding something.
I get what you mean, but actually, our bodies usually handle this balance well. We get potassium from foods like bananas, potatoes, and spinach. As long as you're eating a balanced diet, the sodium-potassium pump works efficiently, and deficiencies are pretty rare.
Yeah, but it doesn't make sense to me how I'm removing poor potassium from my blood. I might as well I don't understand why we do this.
I see your point. The body uses the sodium-potassium pump to keep cells functioning properly. It doesn't actually deplete potassium from your blood. It just shifts potassium into cells while removing sodium. This balance is vital for processes like nerve impulse transmission and muscle contractions. As long as you have a normal diet, your potassium levels in the blood stay stable.
I don't understand how they stay stable. Aren't they supposed to go into the piss?
Some potassium does get excreted in urine, but the kidneys regulate this. They adjust how much potassium is reabsorbed or excreted based on your body's needs.
But we're getting it in. So in order to get it in, to get sodium out, it means it's going to be peed, no?
Exactly. The body is pretty efficient at balancing this. Potassium enters cells, and if there's excess, the kidneys help remove it through urine. So while the pump moves potassium into cells, any surplus can be excreted, keeping your blood levels stable.
So it's going to be absorbed and then go out again?
Right. The body constantly fine-tunes potassium levels. It gets absorbed into cells as needed, and any extra gets excreted by the kidneys. This way, potassium stays balanced in your blood.
Seems so inefficient.
It might seem that way, but it's actually constant balancing act ensures cells function optimally and prevents harmful imbalances. Plus, the kidney's ability to excrete excess ensures we don't accumulate too much potassium.
I don't get it.
No worries. Think of it like a thermostat. The body constantly adjusts potassium levels to keep everything stable.
You know what? Let's keep it. Let's get back to it. So if we go back to the liver, I want to talk a bit about the impressions. So we have a gastric impression and a suprarenal impression and a renal impression. And can you talk to us about all of these and why they're positioned the way they are?
Absolutely. These impressions are formed by organs pressing against the liver. The gastric impression is from the stomach, the renal impression from the right kidney, and the suprarenal impression from the right adrenal gland. These impressions show how closely these organs are positioned to the liver.
Interesting.
Yeah, it's fascinating how these impressions reflect the liver's relationship with nearby organs.
Can I ask about the porta hepatis?
Of course, the porta hepatis is the gateway to the liver. It's located on the underside and allows structures like the hepatic artery, portal vein, and bile ducts to enter or exit. It's a crucial area for liver function and blood flow.
Hepatic ducts, hepatic portal vein. What is all of this?
So, the hepatic portal vein brings nutrient-rich blood from the intestines to the liver. The hepatic artery supplies oxygenated blood. The hepatic ducts carry bile from the liver to the gallbladder and intestines for digestion. All these structures pass through the porta hepatis.
Interesting. So let's talk a bit about the ligaments and the bare area. What's the bare area in the liver?
Sure, the bare area of the liver is a section on the posterior side where the liver is in direct contact with the diaphragm without any peritoneal covering. Ligaments like the coronary and triangular ligaments help define this area. It's important because it provides a route for infections or tumors to spread between the liver and diaphragm.
Tell me one more time.
The bare area is on the top back side of the liver. It's where the liver touches the diaphragm directly. Think of it as a small patch with no protective covering. The coronary ligaments form the borders.
Interesting. So we have a right triangular ligament, and then, okay, we have also a left triangular ligament.
Exactly. The right and left triangular ligaments are formed where the coronary ligaments meet at the edges of the liver. They help anchor the liver to the diaphragm. The right triangular ligament is on the right lobe, and the left one is on the left lobe. Yeah, they basically mark the upper corners of the liver, keeping it stable.
And we have a posterior fold of coronary ligament. What's that?
The posterior fold of the coronary ligament is part of the peritoneal reflection that attaches the liver to the diaphragm. It runs along the posterior surface, helping to define the bare area and stabilize the liver in place.
I don't understand the location of the coronary ligaments, to be honest.
No worries. The coronary ligaments are on the top side of the liver, attaching it to the diaphragm. They form a crown-like shape around the bare area. The anterior and posterior folds come together to form the right and left triangular ligaments.
Let's see if Wikipedia can help.
So the coronary ligament has two layers, anterior and posterior, connecting the liver to the diaphragm. These layers come together at the edges, forming the triangular ligaments. This helps anchor the liver in place.
Okay, so I think I have to start actively recalling the information you're saying and making sure that I'm actually saying it correctly, and also that I'm, not just memorizing it, but actually understanding it, which is a tricky thing, isn't it? How do you know that you actively recalled something, but you did it the right way? You did it by actually understanding it and not memorizing it.
It can be tricky. One good way to check understanding is to explain it in your own words, or teach it to someone else. If you can do that without relying on exact definitions, you're likely understanding it. Also, applying the concept to different scenarios helps solidify it.
Okay, so basically, if we have the visceral view of the liver, then we have a right and left triangular ligament, and we have the left on the left side of the liver, and the right on the right side of the liver, and then we also have the posterior fold of the coronary ligament, which I don't really understand what it does, if you could help me out. And we also have the coronary ligaments, which I don't understand what their function, to be honest. And we have the bare area, which I don't really understand its location.
All right, let's break it down. The coronary ligaments run along the top edges of the liver, connecting it to the diaphragm and forming a crown-like shape. Their job is to anchor the liver. The posterior fold helps define the bare area, which is that uncovered part of the liver in direct contact with the diaphragm. The right and left triangular ligaments are where the coronary ligaments meet at the sides.
And okay, fair enough. Okay, let's talk a bit about the lesser sac now. Is that okay, lesser sac? Oh, it's being shy, guys. Okay. ChatGPT, can we talk a bit about the lesser sac?
Sure. The lesser sac, also known as the omental bursa, is a cavity in the abdomen. It's located behind the stomach and the lesser omentum. It communicates with the greater sac, which is the main part of the peritoneal cavity, through an opening called the epiploic foramen. This space allows for the movement of the stomach and other organs.
I want to understand its anatomical location a bit more.
Absolutely. The lesser sac is situated behind the stomach and the liver. The front boundary is formed by the stomach and the lesser omentum, while the back boundary includes the pancreas. The epiploic foramen, which connects it to the greater sac, is like a small doorway bordered by the liver, the duodenum, and a vein called the inferior vena cava.
So tell me again how it connects with the epiploic foramen.
Sure, the epiploic foramen, also called the foramen of Winslow, is the opening that connects the lesser sac to the greater sac. It's bordered in front by the free edge of the lesser omentum, which contains the chordal vein, hepatic artery, and bile duct.
I'm a bit confused. Break it a bit more simply, because it's a bit of a hard topic for me.
No worries. Think of the epiploic foramen as a small door.
Oop. Where'd you go, dude? Let's ask again, boys. So if you could tell us again how the epiploic foramen is connected with the lesser sac?
Sure. The epiploic foramen, also known as the foramen of Winslow, is the opening connecting the greater sac, which is the main part of the peritoneal cavity, to the lesser sac, which is the smaller recess behind the stomach. It's bordered by the liver, the hepatoduodenal ligament, and the inferior vena cava. This foramen allows communication and the passage of structures like vessels between these two spaces.
I'm confused.
No worries. Think of the epiploic foramen as a doorway. It connects the main area of your abdominal cavity with a smaller space behind the stomach. This doorway is surrounded by the liver, a ligament with blood vessels, and a large vein. It's like a hidden passage in your abdomen.
So it's a passage that passes to what? And why is it called the epiploic foramen? Explain to me how its name relates to its structure, function, location, or whatever.
Sure. The term epiploic comes from epiplone, which is an old term for the omentum, a fatty layer that drapes over the intestines. Foramen means an opening. So the epiploic foramen is named for its location near the omentum and its function as an opening between the greater and lesser sacs in the abdomen.
Okay, so it's an opening between the greater and the lesser sac. Okay. Interesting.
Glad you found it interesting. The body's full of these little hidden details.
Okay. Give me 1 second, guys. Can I ask, what is environmental physiology?
Absolutely. Environmental physiology studies how organisms adapt to their physical surroundings. It looks at how things like temperature, altitude, and humidity affect living beings. This field is especially important for understanding how plants and animals survive in extreme environments.
Interesting. And what
It's a
do we
fact.
know about renal function in the field of environmental physiology?
In environmental physiology, renal function is key for understanding how animals maintain water and electrolyte balance in different environments. For instance, desert animals have highly efficient kidneys to conserve water, while freshwater fish have kidneys that excrete excess water. It's all about adapting to available resources.
What about heat on the renal function?
Heat can stress renal function because the body works harder to cool down, often leading to dehydration. The kidneys then conserve water by concentrating urine. In extreme heat, this can strain the kidneys, highlighting the importance of staying hydrated.
Interesting. Okay, let's go back and talk a bit about the basic renal process, specifically tubular secretion.
Sure, tubular secretion is when the kidneys move substances from the blood into the urine within the renal tubules. This process helps eliminate waste products, like hydrogen ions and toxins, and helps maintain acid-base balance. It's essential for keeping blood chemistry stable.
Can we talk about the 2 kinds of pumps, perhaps?
Absolutely. There are 2 main types of pumps involved in tubular secretion. Primary active transport pumps, which use energy directly from ATP, and secondary active transport pumps, which rely on ion gradients. Primary pumps directly move ions like sodium, while secondary pumps use the gradient created by primary pumps to move other substances.
Interesting. What do you think is the most important job in the world? And I know there is not one answer, but if you had to choose, there's a gun in your head, you have to choose or you're gonna die.
Haha, that's quite the hypothetical. If I had to choose, I'd say anything that directly saves lives, like a healthcare worker. But honestly, every job has its own vital role in society.
Yeah, but dude, what about if you can save, like, politics? Maybe you will save 1 million lives if you stop war.
That's a great point. Political roles can definitely have a huge impact on a global scale. Leaders and policymakers can shape the future and potentially save countless lives by promoting peace and social justice.
Yeah. And I guess you're not necessarily like limited to only one thing, are you? Like, if you see the founding members of the United States, which were they? Ben Franklin, and which are the other ones? Thomas Edison.
You're right. People like Benjamin Franklin and Thomas Jefferson weren't limited to one field. Franklin was a polymath, inventor, writer, and statesman. Jefferson was also a statesman, architect, and philosopher.
So they did a lot of things, you know, like, and I don't know. I mean, I know we live in this specialized age, but
Yeah, it's true. We live in a specialized age, but there's still room to explore multiple interests. Being well-rounded can be an asset in many fields.
yeah. Okay, interesting. So,
Glad you found...
okay, and so you said we have active transport and secondary transport, primary and secondary. I wonder, do we have different for organic acids or acidic drugs like uric acid and different for organic bases or alkaline drugs? How does it go?
Yes, we do. For organic acids like uric acid, there are specific transporters in the renal tubules that handle their secretion. Similarly, organic bases or alkaline drugs have their own set of transporters. These systems help the kidneys efficiently manage different substances.
Do we have such a thing as inorganic acid?
Yes, we do. Inorganic acids like hydrochloric acid are also present in the body, especially in the stomach for digestion. The kidneys help regulate inorganic ions, like hydrogen ions, to maintain acid-base balance.
Interesting. Okay, I'm gonna go a bit in a different angle now, and I'm gonna ask you a bit about lymphatic drainage when it comes to the liver.
Sure, the liver has an extensive lymphatic drainage system. It helps remove excess fluid, proteins, and waste products from liver tissues. This lymph then drains into the thoracic duct, eventually rejoining the bloodstream. It's crucial for maintaining liver function and overall fluid balance.
And we have a superficial one and a deep one.
Exactly. The liver's lymphatic system has both superficial and deep networks. The superficial lymphatics drain the liver's capsule, while the deep lymphatics handle fluid from within the liver tissue itself.
And we have
Both
a
systems work.
hepatic, a celiac, a cisterna chyli. Could we talk about all of the superficial and deep ones and explain to me why they're classified as superficial or deep?
Sure. Superficial lymphatics are just under the liver's capsule, handling fluid from the liver's surface. The deep lymphatics are within the liver tissue, draining fluid from the internal structures. Both eventually lead to the hepatic lymph nodes, then to the cisterna chyli, which collects lymph before it enters the thoracic duct. They're classified this way based on their location relative to the liver's surface.
Interesting.
Glad you think so. Anything else you're curious about?
Well, I'm just wondering, like, why these specific lymph nodes and ducts are the ones that are, like, the thoracic duct is both superficial and deep. How is this possible?
The thoracic duct runs deep within the chest, but collects lymph from both superficial and deep lymphatic vessels. It serves as the main channel for lymph to return to the bloodstream, making it a crucial part of the lymphatic system. Its deep position protects it while allowing it to gather lymph from all over the body.
Can you explain to me how cisterna's chyli's name is related to this structure and function?
Sure. The cisterna chyli is a sac-like structure that serves as a major lymph reservoir. Its name comes from the Latin word cisterna, meaning tank or reservoir, and chyli, referring to chyle, a milky fluid rich in fats absorbed from the intestines. It collects lymph from the lower body before it moves into the thoracic duct.
And why is it superficial and not deep?
The cisterna chyli is actually located deep in the abdomen, near the spine. It's not superficial. It's well protected internally. Its location ensures it can effectively gather lymph from the lower body before channeling it upwards.
And what about the hepatic and celiac? Why are they superficial?
The hepatic and celiac lymph nodes are located near the liver and major abdominal arteries. They're not exactly superficial either. They're positioned deeper in the abdominal cavity. They help filter lymph from the liver and surrounding organs.
What
Their classification.
about the posterior mediastinal lymph and the phrenic lymph nodes? Why are they deep?
The posterior mediastinal and phrenic lymph nodes are considered deep because they're located within the thoracic cavity. They handle lymph drainage from the lungs, diaphragm, and other deep structures. Their position deep in the chest protects them and allows them to efficiently process lymph from these critical areas.
What about the anterior mediastinal lymph node? And how come the mediastinal comes to the liver?
The anterior mediastinal lymph nodes are also located within the thoracic cavity, but in front of the heart. They're considered deep because of their position. As for connections to the liver, lymphatic drainage can involve pathways that connect various abdominal and thoracic structures, allowing lymph to flow between regions.
Interesting. Let me ask you a bit about the liver's postnatal circulation then.
Sure. After birth, the liver's circulation changes significantly. The umbilical vein closes, and the liver relies on the hepatic artery for oxygen-rich blood and the portal vein for nutrient-rich blood from the intestines.
So the
This
umbilical vein closes, you said, step by step.
Sure. Right after birth, the umbilical vein closes as it's no longer needed. The ductus venosus, which used to bypass the liver, also closes. Blood flow then shifts entirely to the liver's own vessels, the hepatic artery and portal vein, for its blood supply.
So how is the ligamentum venosum and the hepatic portal vein and the ligamentum teres involved?
The ligamentum teres is the remnant of the umbilical vein. It runs from the umbilicus to the liver. The ligamentum venosum is the remnant of the ductus venosus, which used to connect the umbilical vein to the inferior vena cava. After birth, the hepatic portal vein takes over, bringing nutrient-rich blood from the intestines to the liver.
Explain to me step by step and don't rush, please.
Of course, after birth, the umbilical vein closes, becoming the ligamentum teres. The ductus venosus, which shunted blood past the liver, also closes, becoming the ligamentum venosum. Now, blood enters the liver.
Wait, so the
via
ductus venosus becomes the ligamentum venosum?
Exactly. The ductus venosus closes after birth and forms the ligamentum venosum. It no longer carries blood, but remains as a fibrous remnant.
Interesting.
It is. The body has all these fascinating adaptations.
Okay, last, can we talk a bit about the portal triad?
Sure. The portal triad consists of three structures: the hepatic artery, portal vein, and bile duct. These are found together in the liver's portal areas, entering and exiting at the corners of liver
So
lobules.
it's the hepatic artery, the portal vein, and
the bile duct. Together, they form the portal triad, crucial for liver function.
I see. And it runs in the free edge of the lesser omentum. I wonder why the lesser omentum.
Yes, the portal triad runs in the free edge of the lesser omentum. This positioning helps it connect the liver to the stomach and duodenum, providing a pathway for blood vessels and bile ducts.
And we also have lymphatic vessels there, or no?
Yes, there are also lymphatic vessels in the lesser omentum. They help drain lymph from the liver, stomach, and surrounding areas. This ensures efficient fluid balance and immune function.
What about nerves from the hepatic plexus?
Nerves from the hepatic plexus also travel through the lesser omentum. They provide autonomic input to the liver, regulating functions like blood flow and bile production.
Okay, I'm gonna call it a day, well, not a day, an episode, and we'll come back for part 3 of this episode in a few hours, really. So yeah, thanks for watching, guys. So part 2 out of 3, and have a good day.